So I would like to just start by echoing the words of, I think, Dr. Jackson earlier, who said that thank you very much to the sRNA for hosting this, and a very special thanks to Carlos Pardo, who I think really does set the tone in the field of AFM study as being a field where people are very collegial and very focused, no matter what angle they're coming from, basic science or patient care, very focused on finding out answers to questions and ways to help kids. So as a pediatrician myself, I think that makes me feel very good. I have nothing to disclose financially, but I am a co-inventor for a patent for some of the antibodies I'm going to be talking about today. And what I want to start with is actually talking about the polyclonal human antibody response to enterovirus D68 infection. So everything I'm going to talk about today has to do with enterovirus D68 infection. As has been covered, there are other viruses that can cause AFM, but we're going to focus on D68 for this talk. And the conclusion, just to give it away right away, is that nearly every adult in the world has neutralizing antibodies floating in their bloodstream to this virus. But there is an age group in childhood that is corresponding with the same age group of kids who get AFM, where there is not this universal presence of neutralizing antibodies. We've isolated a number of human monoclonal antibodies, so now moving into the individual antibodies that make up a human antibody response to infection. And some of those antibodies neutralize the virus quite well in vitro, but very few of them neutralize viruses across all the different clades. So that's reflecting on, especially what Dr. Schuerman talked about earlier, how there's different types of the virus that circulate. One of these antibodies that is quite cross-reactive works very well in an in vivo mouse model of infection, similar to what Dr. Tyler talked about earlier. And we are trying to develop that antibody for use in humans. So I'll give you an update on where we are with that. So just to begin, when I started trying to understand and study the antibody response to this infection in humans, as I said, it was easier to see what was known about the polyclonal antibody response. So these are seroepidemiology studies. I'm going to show you 3 of them. This is the first of 3 slides. And they make points that are reflected in a number of other studies that I won't talk about today. This particular slide shows that adult humans, in this case pregnant women, have been making neutralizing antibodies to this virus essentially across the board since at least the 1980s, as has been shown in the column on the far left, and that even looking across these many decades, there's very few people who do not have neutralizing antibodies to the virus in their serum. So I've represented that with just I've shaded this as red. Now, when we look at a study that includes some childhood age groups, this is looking now in the UK, you can see on the left in 2006, there is an age group of about a half a year to 1 year old where about a third of those kids did not have neutralizing antibodies. And then as they got older, you could see that most of those kids did have neutralizing antibodies. And that was before they had a major enterovirus D68 outbreak. And then in 2016, after a D68 outbreak had happened, you can see that most people did have neutralizing antibodies, but it was still that same age group where there was this incomplete presence of antibodies. And this is one of my favorite studies because it really zooms in on pediatric age cohorts. When children are born, so brand newborn babies on the far left, they all have neutralizing antibodies in this study. And that's probably because they've just received their bolus of maternal antibodies transplacentally from their mother. And you can see that by about 1 year of age, almost none of these children had neutralizing antibodies to D68. And that was incomplete through the course of these first early years of life, these early years of childhood, leading into the school years. But by the time they were in their preteen and teenage years, it was almost as if they were adults where nearly everybody had neutralizing antibodies. So as I said, those are just 3 examples of a number of seroepidemiology studies that show that really nearly every adult in the world, and this is true across the world, this is true across time, has neutralizing antibodies to enterovirus D68 circulating in their bloodstream on a polyclonal level. Newborns also sort of have that same phenotype, but there is this age group, those early years of childhood, where not everybody has these neutralizing antibodies. And that's a very similar age window for what we see as the patients who get acute flaccid myelitis. So putting this together, it led us to this hypothesis that's not terribly revolutionary, but just the fact that antibody probably protects from acute flaccid myelitis, although not from enterovirus D68 infection, because we know from poliovirus vaccinology that polio vaccines, especially the inactivated polio vaccine, make great neutralizing antibody responses. They don't really prevent poliovirus infection in the intestines they're better than not being vaccinated, but they certainly don't prevent it. But they, across the board, really do a great job of preventing poliomyelitis, which is really just AFM due to the poliovirus. So we figure this is probably a very similar case for AFM due to enterovirus D68. And to try to study this, we just wanted to catalog what are the different types of individual monoclonal antibody within those polyclonal responses that humans make in response to enterovirus D68 infection, and then how do those antibodies modify pathogenesis. So in the Crow Lab, the way we do this is we use a human hybridoma technology where we obtain blood from people who've had a particular immunogen or viral infection. So in this case, Jim Gurn at the University of Wisconsin had followed a cohort of children who back in 2014, during that sort of initial large enterovirus D68 outbreak, these children had respiratory disease. He obtained their blood about 3 years after that in 2017 and sent it to us, where we could use the memory B cells in that peripheral blood, infect them with Epstein-Barr virus or EBV, which transforms the cells, causes them to proliferate very rapidly. So this is what you see in this cell culture dish here. These are just colonies of rapidly proliferating B cells. Those secrete a lot of antibody. And then we can use an assay to try to find cell culture supernatants that contain antibodies of interest. So in this case, we used live virus that was coated onto an ELISA plate. And then if that antibody, as would be represented in blue here, was present in the cell culture supernatant, we could then detect that antibody with a fluorescent substrate and a reporter secondary antibody. So we identified 62 of these monoclonal antibodies. And one of the first things we did was just determine how many of these antibodies can neutralize the virus in vitro, in the laboratory, in a cell culture dish. This is a pretty simple assay called a CCID50 assay we do, where you just mix various amounts of antibody with a fixed amount of virus. And then after letting that mixture incubate for an hour or two, add the mixture to a cell culture monolayer. And then a few days later, you just fix that cell culture monolayer, stain it, in this case with crystal violet. And what you can see on the left side of the plate here, where the antibody concentrations are the highest, those monolayers are intact, so virus was neutralized. And then as the antibody is diluted from left to right across this plate, you can see at some point the antibody is diluted to where you can no longer neutralize the virus. And this allows us to, as a measure of the potency of antibody neutralization, we generate this value called an IC50, or 50% inhibitory concentration. That's what's represented in this graph on the right, where each of these individual points is one IC50 from one monoclonal antibody. And I've shown about the 20 or so most potently neutralizing antibodies. And that scale of potency is represented on the y-axis. So higher up on that y-axis is a more potently neutralizing antibody. And some of these antibodies, like this 228, which you're going to hear about in the coming slides, do neutralize very strongly. And they neutralize all the different types of virus we tried. So that Firmon is the one from way back in the 60s. And clade B1 and clade D, those are some of the modern circulating clades of D68. However, it's actually most of these antibodies, including our most potently neutralizing antibody, that really only neutralized the clade of virus that these kids had. So we know these kids all had clade B1 infections, and most of the antibodies really didn't cross-react well to the other strains of virus. So when we were trying to think what might make a good therapeutic, obviously we wanted an antibody that was going to cross-react well amongst different virus clades. So we focused on this 228. We came to the NIAID, specifically DMID's preclinical services for researchers. Yoon Chung Park is the PO, who then hooked us up with Bart Tarbett and Brett Hurst at Utah State University to put these antibodies in mouse models of infection to try to see if they might have any modification of pathogenesis in the mice. And I'm only going to show you the neurologic data today, but we also did this with a respiratory model of infection. This mouse model of infection is very similar to what Ken Tyler described before, except that these mice are interferon, type 1 and 2 interferon deficient mice. But the principles are the same, where if they're given an intraperitoneal injection of virus, they develop a flaccid paralysis and will eventually succumb to their infection. And what we did with this mouse model is we gave them antibody. In this case, I'm showing giving the antibody before infection, but we can also, I'll show you data where we give the antibody after infection. And the antibodies we gave are IVIG, because as Dr. Tyler had shown, IVIG can definitely be protective in this mouse model of infection. We gave our antibody, and then we also gave a placebo antibody that's an RSV antibody similar to palivizumab, for example. And when we gave the antibody as prophylaxis, so prior to infection, we saw that it had a benefit in both survival and neurologic scores. So the way the data is shown here, the data sets that are in black, those are the mice that were given placebo. Those mice universally succumbed to infection by day 7, and those mice got very sick. So then this neurologic score is a higher score on the y-axis is a sicker mouse with more paralysis. Mice given IVIG, those mice certainly did better. So they definitely had a survival benefit in this model. They had less sickness, less paralysis. But the mice given our monoclonal antibody at equivalent doses to the IVIG and even 10-fold lower doses than the IVIG, all of those mice lived. None of those mice got sick at all. When we looked at the viral titers in their blood, what we saw was that there is a viremia for the mice given the placebo antibody. Mice given IVIG certainly have less viremia, but there was no viral replication whatsoever that we could detect, no living virus in the bloodstream of these mice. And this was just a very exciting finding because really there's so many talks from before that this applies to, where this shows that vaccinology should very well, as we all suspected, but should really work very well. If there's neutralizing antibodies present in the bloodstream, it should very well protect from acute flaccid myelitis. At least we would hope that this would extrapolate to humans in that way. But also this antibody could potentially be used for immunoprophylaxis if there was no vaccine. And I will point out that if you're wondering why is this data a little bit different from Ken Tyler's, that's because Ken used doses of IVIG that were much more like what we would give to humans, so about 2 grams per kilogram. And obviously this is 10 milligrams per kilogram. So these are orders of magnitude less IVIG being given in this experiment. So that is an important distinction. Now what happens when we give as treatment? So very similar to what Ken showed before is it's time dependent, but treatment does work. So in this experiment, the way I've shown it is, again, the placebo-treated mice are in black. They all succumb, in this case, by 6 days after infection. We give antibody at different time points after infection, which are indicated by the different colored arrows. And then the data set of the same arrow color is the corresponding data set. So mice given antibody 24 hours after infection, they all lived. 48 hours after infection, there is indeed a significant survival benefit. And then even 72 hours after infection, this one mouse lived. Now that's not a statistically significant improvement in survival. But what's really important is when you look at these neurologic scores, that same sort of light purple line, that mouse that lived had some paralysis and got better. And then when we go back and look at these other treatment groups, there are multiple instances of a mouse that had some paralysis and got better. And this really, again, goes with what Ken talked about with the sort of mouse-human chimeric antibodies that they had done these similar experiments with, where the antibody was given after sickness. So this is just another example of that. And then when we look at the viremia, the bottom line here is within 24 hours after an antibody being given, there's no longer any circulating virus in the bloodstream of these mice. So this, we were very fortunate to be approached by Zab Bio and their partners at Kentucky Bioprocessing to try to make this antibody in GMP, or good manufacturing practice quality, which is basically to say that can go into humans. They've been doing this pro bono, so this has just been a team effort across industry, academia, and really a lot of good partners at NIAID have been helping along with this as well. Where we're at right now is this antibody has been made at GMP. It's actually made in tobacco plants. We've shown in vitro that this antibody works just as well as the antibody we made in our lab. And then actually just 2 days ago, so hot off the presses, we've shown that in vivo, the antibody also behaves exactly as what I just showed you before with the antibodies made in our lab. And so we're close to embarking on studies of making sure the antibody doesn't cross-react to human tissue, making sure the antibody is safe in rat toxicity studies. These sort of very traditional steps we take with hopes to be in phase 1. Potentially as soon as this year, honestly, is it realistic? And then, who knows what's going to happen with the epidemiology of norovirus D68 with all of our social distancing measures. I mean, we all expected there would be an outbreak this summer. I think we all still are very much on guard for this and very aware for this, but it could be that we don't have another outbreak till the next summer. And it is very possible we could be ready to be poised for phase 2 trials by then. So in summary, nearly every adult in the world has neutralizing antibodies to norovirus D68 in their serum. There is an antibody-naive cohort, and that age cohort overlaps with the AFM age cohort. Humans are capable of making antibodies that are cross-reactive amongst different norovirus D68 clades, even on the monoclonal level, which suggests, some of this work suggests that then vaccines will hopefully be able to prevent AFM, and that this could even be a therapeutic measure. And so just in closing, I'd really want to thank, there's too many people to thank everyone individually, but especially our funding sources. So the Pediatric ID Society, St. Jude Children's Research Hospital funded my fellowship for the past 3 years. And then at NAID, both Joe Breen and DAIT gave us a supplement to a grant that has funded a lot of this research. And then, as I said before, DMID, especially Yun Chung Park, has also really been very helpful to us. So thank you to all of those supporters. And I guess this is a good time to transition into our next talk. So Dr. Ann Ye from Sick Kids in Toronto will tell us about AFM from the clinic to the bench and back to the patient.